Right, today we're going to have a look at the second half of protein synthesis. Previously we talked about transcription, so that's where the genetic code in the form of DNA was initially transcribed into pre-mRNA, and then the introns were spliced out of the pre-mRNA strand, resulting in messenger RNA. If we just write in then the corresponding mRNA strand, so we're going to have a U, G, 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 G. We're going to have A, U, G, and then finally U, C, C. The important thing about translation is that we're going to look at another type of RNA molecule, which is called transfer or tRNA. It's still an RNA molecule, so it's made from a single polynucleotide strand, but along the length of the polynucleotide strand, bases complementary base pair. That's what these lines just represent, hydrogen bonds. So you've still got a single polynucleotide strand, but it forms this unusual, and it's said to be a clover leaf shape. The two key things about a tRNA molecule that make it important for its function is that at the bottom of the tRNA molecule you have a sequence of 3 exposed bases, and these are, or this is referred to as the anticodon. Just as we saw previously when talking about the genetic code, because it's all based on the sequence of 3 bases, there are a total of 64 possible combinations of bases, whether it's triplet code in DNA or the corresponding codon in mRNA. We now have a corresponding anticodon in tRNA. So there are 64 different possible anticodons, and each one of those anticodons, so if I give the example of C, anticodon, what that means is that that specific tRNA molecule with the anticodon of C means that at the top of the tRNA molecule you have a binding site, and this is where a single one type of amino acid is going to be carried. And so in this case it would be glycine. So effectively this is where the genetic code actually results in a physical, tangible polypeptide being synthesized. So I'll say again, there are 64 different possible anticodons, and therefore there are 64 different tRNA molecules, and a tRNA molecule with one anticodon type will only bind with and will only carry one specific type of amino acid. Typically, this is where you're going to get questions where you've been given an mRNA codon table. So you've got the 64 different mRNA codons showing you the corresponding amino acids that they're going to code for, and you might be asked to work out the sequence of the DNA triplets from the mRNA code, or you might be asked to work out the sequence of amino acids from the mRNA sequence, or tRNA. That whole combination of different sorts of questions that you might get. So if you have your mRNA codon table to hand, we can think about the sorts of things that you might be asked. Right. So if we start then, the whole process, and this is going to be slightly Blue Peter, so we'll see how we get on with these little models that I have made. Let's remind ourselves at the end of transcription, where you've now got the mRNA molecule that is small enough to move out of the nuclear pores and then to ribosomes. We know that protein synthesis takes place at ribosomes, so this is a ribosome that I've had a go at making. Here's the mRNA molecule, and what will happen is that the ribosome will bind with the mRNA molecule itself. We know that ribosomes are made up from protein and another type of, the third type of RNA that we need to know about, which is ribosomal RNA, and it forms this structure. So the ribosome binds with the mRNA molecule, and it binds so that you can fit 2 codons or 6 bases, so 1, 2, 3, 4, 5, 6. You can fit 2 codons or 6 bases of the mRNA into the ribosome itself. Right, remember, so this is all taking place at the ribosomes, typically at rough endoplasmic reticulum. One of the key things to consider is how does the ribosome know which end of the mRNA molecule to start the process of translation with, and this is where we can use our mRNA codon table. If you find on your mRNA codon table AUG, you'll see that it says start / met or methionine. So the mRNA sequence of AUG, if it is at the beginning of the mRNA molecule, this is what's referred to as a start codon. This doesn't actually result in the mRNA molecule coding for a particular amino acid. It's literally just telling the ribosome to start the whole process at this end as opposed to the opposing end. So initially you have the start codon, and then the process begins. So if we think then that the next mRNA codon sequence is GGG. Let me just stick up here, we have UAC, and that corresponds with start. So if we consider the mRNA codon at GGG, it's going to be complementary to the tRNA codon sequence of CCC. So if we put that in there. So the mRNA codon sequence of GGG means that, and we have a tRNA molecule which is attached to, which has rather an amino acid attached to the top of it. The tRNA molecule brings and carries the amino acid glycine to the ribosome, and this starts off the whole process of translation. What happens next then is that the ribosome will then move along another three bases, or one codon's worth. This is where we'll see how it all works out. So it moves 3 codons along so that we have another, what's referred to as a vacant slot of mRNA and the next codon. Therefore, the next tRNA molecule, which is going to be able to complementary base pair with the next mRNA codon. So if we think that at the next mRNA codon sequence of AUG, so I've chosen this on purpose, it's the same as AUG, but where you find the mRNA codon of AUG within the mRNA molecule and not at the start, it is going to code for methionine. So this is going to have the tRNA codon, sorry, the tRNA anticodon is going to be UAC, so we've got UAC, and the tRNA molecule with its anticodon, complementary base pairs, we can use that phrase again, complementary base pairs with the mRNA codon of AUG. Now at this point, we've got 2 amino acids next to one another. And if we're going to be synthesizing, if the whole point of this is to synthesize a polypeptide, then in order to join those 2 amino acids together, we need a condensation reaction to occur. So enzymes are responsible for catalyzing the condensation reaction resulting in a peptide bond. That needs energy from the hydrolysis of ATP, and what we get then is a peptide bond occurring between those 2 amino acids. So bear with me here. So we get a peptide bond forming between those 2 amino acids, and they join together. So we've now got glycine bound onto methionine. What happens at this point then is that the first tRNA molecule actually detaches from the glycine, and that can return back to the cytoplasm. It can pick up another specific amino acid, and the process just continues and repeats. The ribosome will move down to the next vacant slot, so we now have the mRNA codon of UCC. So we've got UCC. The tRNA anticodon that's going to be complementary there is AGG. tRNA molecule moves complementary base pairs with the mRNA codon of UCC. Again, we've got 2 amino acids next to one another. Enzyme catalyzes the condensation reaction now between methionine and serine, so we get another peptide bond. There we go. The second tRNA molecule returns back to the cytoplasm. It can pick up another amino acid, and this process just repeats itself along the whole length of the mRNA molecule. Obviously we've got a very short one here, and what that results in ultimately is the formation of a polypeptide. That process will continue until right at the end of the mRNA molecule, there's what's called a stop codon. So this tells the ribosome where to stop the whole translation process. Just to remember that ultimately what happens next to that polypeptide is that it can move into Golgi body, where it can be modified, it can be made into a glycoprotein, a whole other series of modification can take place. And finally, just remember that a protein may consist of one polypeptide, or it may consist of more than one. So just remember that a protein such as hemoglobin is made up from 4 separate polypeptide chains, and therefore it is coded for by 4 separate genes. So just to finish off, if we look at our sequence here, the DNA triplet code of CCC becomes the mRNA codon of GGG, which becomes the anticodon of CCC, and that tRNA molecule is specific to carrying glycine, and so on and so forth. And that is the end of translation.